An autosomal dominant mutation is a single mutant allele from an affected parent that is transmitted to an offspring regardless of sex. That's assuming 100% penetrance. There is no carrier, and the affected parent has 50% chance of transmitting the disorder to the child through segregation of chromosomes during gametogenesis. Homozygous dominant mutations are usually lethal. Many are de novo, meaning a spontaneous mutation occurring, and lead to decreased reproduction of the individual, and oftentimes they don't reproduce and don't pass on the mutation to the next generation. However, if the mutation is in a regulatory protein involved in metabolic pathways or key structural proteins, it will likely cause death. Autosomal recessive mutations are manifested only when both members of the gene pair are affected. That is, must be homozygous recessive. You can have carriers, meaning one mutant copy but no diseased phenotype. X-linked recessive mutations are always associated with the X chromosome. The inheritance pattern is predominantly recessive. That means females need both X chromosomes to have a mutation, in order to have the phenotype. In general, if a mutation occurs in a single gene, either one or both alleles, the outcomes are formation of an abnormal protein or decreased production of a protein, even maybe no production of the protein. And an abnormal protein doesn't mean that it doesn't work at all. It might have a new activity in the cell, often referred to as a, quote, gain of function, unquote, mutation. I will highlight a few single gene disorders on the next few slides. Autosomal dominant mutations in the FBN1 gene cause Marfan syndrome. The FBN1 gene provides instructions for making a protein called fibrillin-1. Fibrillin-1 attaches or binds to other fibrillin-1 proteins and other molecules to form thread-like filaments called microfibrils. Microfibrils become part of the fibers that provide strength and flexibility to connective tissue. Additionally, microfibrils store molecules called growth factors and release them at various times to control the growth and repair of tissues and organs throughout the body. A mutation in the FBN1 gene can reduce the amount of functional fibrillin-1 that is available to form microfibrils, which leads to decreased microfibril formation. As a result, excess growth factors are released, and elasticity in many tissues is decreased, leading to an overgrowth and instability of tissues. Because the aorta is considered to be an elastic artery, it should not be a shock why these individuals have a problem with aneurysms. Marfan syndrome is a disorder that affects the connective tissues in many parts of the body. Connective tissue provides strength and flexibility to structures such as bone, ligaments, muscles, blood vessels, and heart valves. The signs and symptoms of Marfan syndrome vary widely in severity, timing of onset, and rate of progression. The two primary features of Marfan syndrome are vision problems caused by a dislocated lens, ectopia lentis, in one or both eyes, and defects in the large blood vessel that distributes blood from the heart to the rest of the body. That would be the aorta. The aorta can weaken and stretch, which may lead to a bulge in the blood vessel wall called an aneurysm. Stretching of the aorta may cause the aortic valve to leak, which can lead to a sudden tearing of the layers in the aorta wall, called an aortic dissection. Aortic aneurysm and dissection can be life-threatening. Many people with Marfan syndrome have additional heart problems, including a leak in the valve that connects the two chambers of the heart, like the mitral valve. It prolapses, or the valve that regulates blood flow from the heart into the aorta, like an aortic valve regurgitation. Leaks in these valves can cause shortness of breath, fatigue, and an irregular heartbeat felt as a skip or extra beats, palpitations. Students miss the connection between valve problems leading to respiratory changes. It is because the lungs try to maintain blood and ventilation matching. We will learn more about this during the semester. Individuals with Marfan syndrome are usually tall and slender from excessive growth factors that aren't regulated, have elongated fingers and toes, arachnodactyly, and have an arm span that exceeds their body height. Other common features include a long and narrow face, crowded teeth, an abnormal curvature of the spine like scoliosis or kyphosis, and either a sunken chest, pectus excavatum, or a protruding chest, pectus carinatum. Autosomal dominant mutations in the NF1 gene cause neurofibromatosis type 1. The NF1 gene provides instructions for making a protein called neurofibromin. This protein is produced in many cells, including nerve cells that myelinate nerves. For example, oligodendrocytes and Schwann cells. Neurofibromin acts as a tumor suppressor, which means that it keeps cells from growing and dividing too rapidly or in an uncontrolled way. Mutations in the NF1 gene lead to the production of a non-functional version of neurofibromin that cannot regulate cell growth and division. As a result, tumors such as neurofibromas can form along nerves throughout the body. It is unclear how mutations in the NF1 gene lead to the other features of neurofibromatosis type 1, such as café au lait spots and learning disabilities. Neurofibromatosis type 1 is a condition characterized by changes in skin coloring, that's pigmentation, and the growth of tumors along nerves in the skin, brain, and other parts of the body. The signs and symptoms of this condition vary widely among affected people. Beginning in early childhood, almost all people with neurofibromatosis type 1 have multiple café au lait spots, which are flat patches on the skin that are darker than the surrounding area. These spots increase in size and number as the individual grows older. Freckles in the underarms and groin typically develop later in childhood. Most adults with neurofibromatosis type 1 develop neurofibromas, which are benign tumors that are usually located on or just under the skin. These tumors may also occur in nerves near the spinal cord or along nerves elsewhere in the body. Some people with neurofibromatosis type 1 develop cancerous tumors that grow along nerves. These tumors, which usually develop in adolescence or adulthood, are called malignant peripheral nerve sheath tumors. People with neurofibromatosis type 1 also have an increased risk of developing other cancers, including brain tumors and leukemia. During childhood, benign growths called Lisch nodules often appear in the iris. Lisch nodules do not interfere with vision. Some affected individuals also develop tumors that grow along the optic nerve. These tumors, which are called optic gliomas, may lead to reduced vision or total vision loss. In some cases, optic gliomas have no effect on vision. Additional signs and symptoms of neurofibromatosis type 1 include hypertension, short stature, an unusually large head, macrocephaly, and skeletal abnormalities such as scoliosis. Although most people with neurofibromatosis type 1 have normal intelligence, learning disabilities and attention deficit hyperactivity disorder, ADHD, occur frequently in affected individuals. Achondroplasia is a form of short-limbed dwarfism. The word achondroplasia literally means without cartilage formation. Cartilage is a tough but flexible tissue that makes up much of the skeleton during early development. However, in achondroplasia, the problem is not in forming cartilage, but in converting it to bone, a process called ossification, particularly in the long bones of the arms and legs. Characteristic features of achondroplasia include an average-sized trunk, short arms and legs, with particularly short upper arms and thighs, limited range of motion at the elbows, and macrocephaly with a prominent forehead. Fingers are typically short, and the ring finger and middle finger may diverge, giving the hand a pronged or trident appearance. People with achondroplasia are generally of normal intelligence. Mutations in the FGFR3 gene, found on chromosome 7, cause achondroplasia. The FGFR3 gene provides instructions for making a protein that is involved in the development and maintenance of bone and brain tissue. Two specific autosomal dominant mutations in the FGFR3 gene are responsible for almost all cases of achondroplasia. Researchers believe that these mutations cause the FGFR3 protein to be overly active. This is a gain-of-function mutation, which interferes with skeletal development and leads to the disturbances in bone growth seen with this disorder.